The question was, what are triple bodies? And they are a new molecular format of 3 antibody-derived binding heads in a single polypeptide chain. And the 2 outer ones bind to a tumor cell, and the central one binds to an effector cell of our choice. This molecular format has been patent protected, and the patents are awarded. And there many examples are possible. You can use different targets on different tumor cells, both leukemias and solid tumors. And our most advanced molecule, our top of the pipeline, is a late preclinical molecule that has the status of clinical candidate selected. That means we have spent about 10-15 years on developing this agent. The first publication was in 2009, and the patent was filed in 2009. And it has a binding site for CD33 and CD123. And those were chosen deliberately, not by accident, because we thought one needed to target AML leukemia stem cells, not only the bulk leukemic cells. And I talked at this meeting in 2004 with Professor John Dick from Toronto, who is a leading expert and a discoverer of AML leukemia stem cells. And we looked for a pair of targets that would be uniquely suited to preferentially target the leukemia stem cells over the bulk. And we agreed that the pair of CD33, CD123 would be good. At that time, we already had the molecular format, and we had already designed several other triple bodies and shown that they worked in an academic setting. So we started to make this triple body CD33, CD16, CD123, where CD16 recruits either NK cells or also gamma delta T cells, and possibly macrophages, we don't know yet. And that molecule was shown to be very effective in cell culture studies with AML-derived cell lines and also with primary material from AML patients. And the first publication on that was in 2010, Kügler and Stein. Our agent at the moment relies on NK cells as the killers. Whereas at this meeting and in the current setting of immunotherapy worldwide, most people rely on T cells. And we deliberately did not want to have the T cells because they generate high toxicity, cytokine release syndrome, and the NK cells are a little more milder. We think the NK cells are intrinsically favorable, although we are also making the same molecule to recruit T cells, and we have published on that. So our triple body format is capable of recruiting T cells. And actually one of the next triple bodies we are currently in the process of making will be the same one that has the same targets, but switching the effector cell recruiting from NK cell to T cell. And then we can directly compare. People now say T cells are better, but if you ask them why, well, they say because NK cells are bad. And I say, where is it published? There is nothing which does a direct comparison between the power of the NK cell and the T cell. So if we get this new agent with a new proprietary T cell binding site, which I cannot disclose, but we have had preliminary work and publications on that. And then we can take from the same patient a bone marrow sample and either add the NK cell recruiting agent or add the T cell recruiting agent and use the autologous tumor cells and the autologous NK cells or the autologous T cells and see which one works better. And we can measure cytokine release, and then we can directly say whether the T cell releases more cytokines than the NK cell.